Soil microorganisms. How to produce them and where to find them. We are a microbiology lab, founded in Ecuador, South America. Our laboratory was named Microbiolab. Here, in our country, we have a great biodiversity, so when we want to find any kind of microorganisms, we go to any of the natural biospheres that we have, but many of these microorganisms are cosmopolitan and can be found in agricultural areas. But they might be possible being extracted from commercial products. Unfortunately, the expected results may not always be obtained, especially if these products are made with microbiological strains extracted in other geographical areas, because they have different crops, different climates, different soils, and different agricultural management. All these factors and the origin of microbiological strains can influence the ability of these microorganisms to function or establish themselves. These imported microorganisms may not establish themselves well in the soil or in crops or perform biological control. They can easily diminish or be useless. It is preferable to try to extract microorganisms from the geographical area adapted to the conditions of said area. There are several methods that can be used to perform these processes. The following methods can be used: microorganism traps, pour plate with selective and differential agars, direct isolation of entomopathogens. Before using any of these methods, remember to take into account the nature of the microorganism you wish to isolate. If it is a fungus, it needs special agars such as potato dextrose. If it is a bacteria, nutrient agar is used. If it's an algae, it also needs special agars and special growing conditions. If they are mycorrhizae, remember that they are microorganisms that grow only in living plant material and cannot be reproduced in vitro. If you perform the extraction of microorganisms from commercial products, it is important to bear in mind that the greater the diversity of microorganisms in the product is, more likely it is to lead to greater contamination. Also, it may be that the microorganism you wish to isolate no longer exists in that product. In liquid media, microorganisms can be unstable because they are living beings. You cannot expect a bacteria to cut its growth curve. The most you can do is lower its metabolism by lowering the temperature of the product without cutting its cold chain from its preparation. If you want to manufacture products that contain mixtures, you have to work with each microorganism separately and mix them when applying. How to produce soil microorganisms? As I already told you, you have to take into account the nature of the microorganism. Each microbial group has its own nutritional requirements and environmental conditions for its development. Basically, for bacteria you need to make liquid productions and suitable bioreactors for their development, respecting their growth curves. In the case of fungi that grow in vitro, you can use substrates such as rice or oats where both their hyphae and spores develop. If you use liquid bioreactors, you can also obtain